Okay, thank you so much. Uh, Hello, everyone. My name is Manuel Fiallos, and actually I'm here to present my research. My research is focused on identification and expression of the AREB, ABF gene family in the Jute under abiotic stress with exogenous ABA. In this form, the next slide is talking about the objectives. The first objective is to analyze the morphological traits of the Jute seedlings with ABA under salt and drought stresses. After that, I will continue, and actually I continue with the identification of the characterization of the gene family from the Jute genome, especially in the species of Corchuris solitorius. After that, I analyze the expression patterns of these genes under salt stress and drought stress with and without ABA. According to the method and the methodology, my research was focused in two parts. First of all, the Jute seedlings was tested under 6 different treatments. How is the control, the ABA treatment, the salt stress. For the salt stress, we use sodium chloride, and after that, we combine with the ABA, salt stress or sodium chloride plus ABA. After that is drought stress. For the drought stress, we use the PAG 6000, and the other treatment is the PAG plus ABA. All of these 6 treatments we tested in different seedlings. Actually, we used around 10 seedlings with a uniform size. In this one, we check the morphological and the physiological changes that is having and actually is suffering in these seedlings. After that, from these seedlings, we took some samples, especially the root, the stem, and the leaf, for checking the molecular analysis. In this form, we extract from these samples the RNA and the cDNA sequences. And this part is the end about this part of the methodology. Meantime, the other one is focused about the genome sequence data. Actually, from this one, we identify the gene family from the Jute genome. We use different bioinformatic tools. How is the blast, the blast from NCBI. We identify the gene structure and the concert motif from the meme. And from this one, when we already identified some genes candidates, we study the cis element, the phylogenetic relationship, and the genetic evolution. When we finish about all of these one, we identify which kind of genes are certain, and actually they are having some functions in our genome. After that, we design some primers, and we combine with the cDNA and actually with the primers, we did the QRT-PCR analysis from the candidate genes from the different samples that we are having or we collected from the different treatments. In this one, we are knowing what is the expression pattern that is having this gene family under salt and drought stress with and without ABA. After that, this one is some of the samples that we are having from our results. And this one is some of the analysis from the bioinformatic tools that we did. After that, the analysis, this one is really important because we need to understand how was working the ABA. Actually, the application of the ABA was at the same time with the stress condition. After that, the movements of the ABA was going up to the plant by the xylem and going down to the plant to the different organs. Actually, this one was the distribution of the ABA and the different nutrients, especially to the roots, by the phloem. And another thing that is really important with the ABA, we have 2 things. How is the systemic stress tolerance and the adaptive response. After that, it's really important to know the different functions that is having the ABA during the application, during the stress conditions. The another thing that is really important to know from this one is the sensor of the ABA receptor, because actually how we are working about the molecular analysis, also is really important to know the relationship that is having the other gene families until to get our gene family, how is the AREB. And all of this distribution, actually this order we are having from our identifications. After that, about the impact that is having our research, especially in this one, I want to invite you, you can check these 2 pictures, because according to the effect of the salt stress, we are having different samples that we took from the different treatments, especially the CK, the ABA, sodium chloride or salt stress, and sodium chloride plus ABA, or salt stress or ST plus ABA. In this form, especially in the application of the salt stress, you can compare with and without ABA. How is the difference with application of the ABA to enhance the resistance of this plant under the salt stress? This one is something very important because actually If we want to compare between the stresses, the drought stress doesn't have so much difference with the application of the ABA. For that reason, all of these treatments, we were talking about some specific doses, because according to this one, we focus that for salt stress with a concentration of 200 millimoles of sodium chloride, we need to work with 10 micromoles of ABA, because if we can increase more this concentration, actually the balance of these chemicals is going bad, because actually the plant will suffer another kind of stress, and this one will die as soon that we are treating. The another thing, according to the drought stress, we are using for 10 millimoles of PHG 6000, we are using 25 micromoles. And this one is the dose that we specify for using in the jute seedlings. Maybe this concentration will vary according to the crop, but especially for the jute, this one was the perfect dose that we are using according to the ABA with the respective stress conditions. This one is the morphological. According to the molecular, we have the expression pattern. We got around of 8 genes from the gene family, but we found that just the gene number 3 and the gene number 7 has the best expression, especially from the leaf and stem. Meantime, the root was not so good, but we still have some expression that we can take it, and actually we can recognize from the graph. After that, from these two genes, we did the identification of the drought stress and the salt stress. Why not do over the other genes? Because we are focused in the expression of the ABA signal in the plant. For that reason, first of all, we run the treatment with the ABA. After that, we choose some genes are specifically in the expression of the ABA in the plant. After that, from this one, we took and to check how is these genes according to the different stresses. And we found it like the gene number 3 and the gene number 7 with the application of the ABA under salt and drought stress has the best expression. And this one is something very important, because under drought stress expression, the highest expression, we can localize in the leaf and the stem. Meantime, in the salt stress, the best expression is in the stem and the root. Something like that is really important, and actually with this one, my research is finished, but in my laboratory is continue the overexpression of these two genes in the jute seedlings until to have the hybridization and to see how is this one the response of these two genes under different stress, not just salt stress and drought stress, but maybe we can check in another stresses from the abiotic characterization. And thank you so much. And this one is all for my presentation.
Thank you, Manuel.